Welcome to our poster presentation. Today we'll showcase how Oaken is harnessing our Multi-Omics Spatial Atlas in Cancer dataset, otherwise known as MOSAIC, for optimizing ADC drug discovery. Antibody-drug conjugates, or ADCs for short, are an increasingly important therapeutic modality in oncology. Over the past decade, there has been a rapid expansion in the number of approved ADCs. These drugs consist of a monoclonal antibody conjugated to a cytotoxic payload via a molecular linker. It's been shown that ADCs can improve efficacy and reduce side effects compared to traditional chemotherapy. Recent innovations include bispecific antibodies, dual payloads, and novel linkers. However, identifying which patients will benefit most from ADC treatment remains difficult. Furthermore, large-scale data-driven approaches to optimize target and payload selection are still limited. In this work, we aimed to quantify the key determinants of ADC clinical success and demonstrate that such analyses can recover clinically validated ADC targets. To address these aims, we used the MOSAIC dataset, a multimodal, multi-time point oncology dataset from more than 2,000 patient samples comprising 6 data modalities: clinical data, histology images, bulk RNA sequencing, whole exome sequencing, single-cell RNA sequencing, and spatial transcriptomics. In our poster, we highlight different examples of how we quantify hallmarks of ADC response. One critical hallmark of ADC response is the homogeneity of target expression. In other words, how evenly it is expressed throughout the tissue. Focusing on Nectin-4, which is a validated ADC target, we illustrate our multimodal approach to quantifying target expression heterogeneity. Our findings reveal differential Nectin-4 expression among cancer cell subpopulations in single-cell RNA-seq data. This observation is corroborated by concordant expression patterns observed across distinct tumor ecotypes in patient-matched, spatially resolved transcriptomics data. We suggest that the observed Nectin-4 expression heterogeneity may impede treatment efficacy. Next, we studied the bystander effect. It's known that cells not expressing the tumor antigen, but in the vicinity of cells that do express the antibody target, can face collateral damage from the payload. This is the so-called bystander effect. Spatial transcriptomics data provides unique insights into this effect. First, we annotate 3 distinct spatial regions: the tumor core, tumor edge, and the stroma. Then, we compute the proportion of tumor spots that either express the target or are direct neighbors of tumor spots that express the target. And we show an example here of ERBB2. This gives us an idea of how much of the tumor we can expect to be exposed to the payload. With this data modality, we also extract feature-dense representations of the immune tumor microenvironment for each patient using a suite of computational biology tools. These representations are then used to predict patient outcomes and inform combination treatment strategies. Now, for some bispecific ADCs, co-expression of the target antigen on the same cells is essential. We therefore developed a method to robustly quantify spot-level co-expression in spatial transcriptomics data using permutation tests. Here we show examples of 2 lung cancer samples with statistically significant co-expression of the EGFR and ERBB3 genes, yet very different effect sizes. This allows for a very deep characterization of co-expression per patient that can potentially inform patient selection for some bispecific ADCs. Next, we illustrate our approach to predict the response of cancer cell subpopulations to different payloads. In recent published work, we trained a deep learning model that we call LEAP. This model predicts the response of cell lines to more than 600 drugs using baseline gene expression profiles. These drugs include several that share a mechanism of action with ADC payloads. We demonstrate that for several relevant drugs, we obtain high performance in cross-validation during training. Testing the LEAP model on patients' single-cell RNA-seq data, we find inter- and intratumor patient heterogeneity in the predicted response of cancer cell subpopulations from bladder cancer patients to a molecule called epothalone B, a microtubule inhibitor, like well-known payloads. As sufficient data becomes available, we will seek to validate the LEAP model in patients treated with ADCs. Finally, we tested whether MOSAIC-derived features can distinguish known ADC targets from other genes. We found that ground truth ADC targets are indeed distinct from other genes according to several features that represent hallmarks of response. We highlight two specific features in particular that were strongly associated with known ADC targets. These were cancer cell specificity and target expression heterogeneity. Future work will scale this analysis to further indications, including the training and benchmarking of more complex target identification models. In conclusion, our study showcases the power of the MOSAIC multimodal oncology data and artificial intelligence in quantifying the biological characteristics that underpin ADC clinical success. By integrating diverse omics data and applying advanced computational methods, we are better equipped to identify and optimize ADC therapies for cancer patients.